everybody. Uh, Welcome to the Society for Immune Polymorphism's 2025 summer webinar. I'm Steve Mack, the Society's president and your moderator for this event. For those of you who are not familiar with the Society, we are an international membership organization dedicated to understanding the genetic and functional variation of the vertebrate immune system and the immune system's role in evolutionary biology, disease, and health. If you're not yet a member of the Society, you can join by going to tinyurl.com/join-the-SIP. That's Society for Immune Polymorphism. And you can find more information at immunepolymorphismsociety.org. You're invited to attend the Society's second annual symposium in Sassenheim, the Netherlands, from November 3rd to 5th of 2025 on polymorphic immune molecules and their functional diversity across species, which is sponsored by the T.X. Miller Foundation. Discounted early bird registration ends on August 15th. You can register and find more information by visiting sip-sassenheim.org. Today's webinar is titled Air and Despair, IGH Duplication in Bats. If you have any questions for our speaker, you can put them in the Q&A or the chat. With that, I'm pleased to introduce our speaker, Professor Hannah Frank. Hannah earned an AB in Organismic and Evolutionary Biology, summa cum laude, from Harvard College in 2009 before studying ecoimmunology in a threatened reptile in New Zealand on a Fulbright fellowship. She completed her PhD in biology in 2017 at Stanford University and did her postdoctoral work in immunology at the Stanford School of Medicine. Hannah's research combines fieldwork, targeted infection screening, genomics, and immune characterization to explore the ecology and evolution of host infection interactions. In particular, she's interested in how host ecology and habitat disturbance impact bat pathogen interactions and the evolution of bat immunity, especially their B and T cell receptor diversity. In her spare time, she enjoys baking, crafting, and tending to her F1s. So please welcome Hannah, and I will vanish and take it from there.
Awesome. Thank you. Thank you all for coming. I'm excited to talk to you guys about bats today. So yes, my title is Air and Despair. That title idea is from Scott Boyd, who I think some of you know. Yes. So anyway, all credit to Scott. And before I start talking about the science, I just want to thank a few of these lovely people. Mostly, I'm going to be talking about my work in collaboration with especially Taylor Purcell, who is a veterinarian, but also a PhD student in Scott Boyd's lab, led a bunch of the work I'm going to talk about, as well as Ashley Rears, my postdoc. Tony Schounts did a lot of the Artibius infection studies. Scott was my postdoc advisor, and he's still a collaborator. And then James Ellison did a lot of the Abscessus infections. So thank you to all of them. Okay, so as Steve said, the two sort of main questions that drive my research are how do host ecology and environment influence infection and host evolution in response to pathogens, as well as how have natural reservoirs adapted to their infections. And so today we're going to focus mostly on this second question, and the immune system, which we all love. Okay, so model organisms are great, and they can provide a lot of useful insights. So on the left here, I have our lab mouse, Mus musculus, has taught us a lot of really fantastic and important biology, but they can't replicate all of the interesting biology that we want to study. So on the right, I have the naked mole rat. These are long-lived species that don't senesce in the same way or get cancer in the same way that a lot of other rodents do. So if you want to study that phenotype, you really have to go to the source. You have to go to the naked mole rat, not the lab mouse. And so what I like to call thoroughly non-model organism are bats. So first, I always put this PSA in because I often am speaking to people who understandably do not think that much about sort of the phylogenetics of mammals. Bats are not flying mice. Mice and humans are actually more closely related to each other than either is to a bat. Bats are most closely related to pigs and whales and cats and dogs. So The next time someone says, aren't bats just flying mice? You can tell them, no, they are flying pig whale dogs. Um, and I'm going to imagine that some of you laughed at my joke. Okay. So why study bats? They are incredibly widely distributed and speciose, and they're a really interesting group of species for a number of reasons. So first of all, there are a lot of them. Um, like many things, they have their center of diversity in the tropics. But it's not just the sheer number of species, but also sort of how ecologically diverse they are. So here I'm showing you one axis of diversity. This is diet. Um, so a lot of bats eat insects, but you also have bats that eat blood. If you're curious, there are actually only three species of bats that eat blood. They all live in Central and South America. So if you're sitting in somewhere temperate right now, they will not be able to get you. Um, and that's out of the over 1,400 species of bats. So very few bats actually eat blood. Some eat nectar. If you enjoy mescal, then you want to think of bat because they are a main pollinator of agave. And all mescal comes from wild agave. There are a lot of fruit-eating bats, thinking about the flying foxes that are in Australia, but also these neotropical fruit bats. And then there are also even fish-eating bats and vertebrate-eating bats. There are bats that eat other bats. This is a fishing one. I think it's just really cool. They fly down, they grab the fish with their feet. Um, okay, so bats are super cool. Lots of really interesting ecology. As a result, they're very important for ecosystem functioning. They do pest control, they do pollination, they do seed dispersal. But as you may be familiar, they also have this connection with a lot of emerging infectious diseases. In particular, they seem to be the asymptomatic reservoirs of a number of these, mainly viral infections. So here you have some people standing outside a Python cave in Uganda, capturing Egyptian rosette bats to test them for Marburg virus, where this is an area that has been linked to Marburg outbreaks in the past. And it's not just filoviruses, including Marburg and we presume Ebola, but also things like sarbecoviruses, SARS-CoV, SARS-CoV-2, merbecovirus, another coronavirus associated with camels in the Middle East, um, as well as paramyxoviruses like Hendra and Nipah. Hendra is a problem in Australia, Nipah is in South and Southeast Asia. And then the one that people in the Americas are probably most familiar with, rabies. So this leads to a question of why do bats have these infections? Why are they so good at hosting these things and not really showing symptoms when they're infected with pathogens that absolutely that are lethal to most other species? And we think that some of this is due to their biology. Because bats break a lot of mammalian rules. They're the only flying mammals in the world. Um, they and with flying comes a lot of trade-offs. So they have to, flying is incredibly metabolically intensive and creates a lot of reactive oxygen species. And so we think that actually it also causes dramatic temperature fluctuations because when you're doing all of that activity and you're an endotherm, you get very, very warm. And then when you stop, you can get very, very cold. So we think that actually these adaptations for flight might aid in their viral tolerance. So both, as I mentioned, flight creates these reactive oxygen species, a lot of free radicals that cause DNA damage, but viral infection can also cause a lot of DNA damage. And so some of these pattern-associated molecular patterns are also the same as these damage-associated molecular patterns. And usually, in most species, when the body senses DNA damage, it leads to inflammation. Um, but in bats, they seem to be very good at tamping down this inflammation. So I have the human over here, and it's like when it senses DNA damage, it unleashes this really angry mob that can be really detrimental to the host, whereas bats seem to be much better at regulating things. So here's a paper from about 10 years ago showing dampen sting dependent interferon and activation in bats. And there seems to be a lot of interesting studies suggesting that bats just don't inflame in the same way as humans and other species. The another thing that is odd and frankly really cool about bats is that they live for a very long time. So in general, the bigger you are, the longer you live. You probably have an intuitive sense of this. Mice don't live as long as elephants do. And so we can quantify this as a longevity quotient, which is basically your observed lifespan over what you would be expected to live given your size. And you can see that most things fall along this line, except then you have bats. Bats are incredibly long-lived for their size. There are species of bats where individuals have been found to live over 40 years, and they're 5 grams. So that's like an anemic lab mouse living to 40. It's insane. And you'll see that of the 19 longest lived for their size animals, 18 of them are bats. The other one is the naked mole rat over here. So the fact that bats are flying, they're not inflaming, they're living a really long time, they don't seem to get cancer, they're hosting viruses that kill people but don't seem to cause them problems, has led to this question and speculation: is bat immunity somehow special? Um, so, as I'm sure you are familiar, immunity can sort of be broken into these large branches: innate immunity, these non-specific, relatively quick mechanisms, as well as adaptive immunity, which is the pathogen-specific arm. Usually takes a little while to develop, but then is really quick upon secondary exposure. Um, and so I'm often talking to ecology and evolution audiences. And who doesn't love a good analogy or metaphor? So if we think about the immune system as, a feudal castle system, um, you've got your innate defenses and your adaptive defenses. And this, there's been these sort of hypotheses that because there are so many bats and so many different viruses, and none of these bats really seem to be dying of them. I will say that rabies can be fatal in bats. Um, that there's an idea that bats have this spectacular innate immunity, and that their adaptive immunity is relatively unimportant. So using this castle metaphor, it seems like humans have this relatively, welcoming castle and this angry mob, whereas bats have this really, really well defended castle with, a community officer telling people to calm down. Um, and there's a lot of evidence that it's true that bats have really interesting innate immunity. I mentioned there's a lot of dampened inflammatory pathways, there's a lot of dampening of interferon stimulated genes. And I myself have done a little bit of this looking at pattern recognition receptors, especially toll-like receptors, as well as some RIG-I-like receptors. And we do definitely see that bats evolved early to detect pathogens, especially viruses. And I'm happy to talk more about this and take questions. I just kind of figured you guys would be more interested in the adaptive stuff. But what about the adaptive stuff? This has historically kind of been ignored amongst people who are interested in bat immunology. So why should we care if they do seem to have all of these interesting innate immune adaptations? This is one of my students drawing a little bit of blood from a bat in the field. I don't have a lot of field pictures in this presentation, so here you go. Okay, so why should we care about adaptive immunity? As you guys know, in general, adaptive immunity is pretty important for fighting pathogens in most species. They're incredibly, especially immunoglobulins, which is what I'm going to focus on. They're a really important component of the adaptive immune system. You guys probably know better than me the myriad functions that they get involved with, whether it's activating complement or opsonizing things or just directly neutralizing antigens, ADCC, any number of both sort of direct and indirect functions of these immunoglobulins. And we know that they're an important thing for bats because people keep doing these wonderful serology studies, right? And they show, serological evidence for Henipa-like and Philo-like viruses in Trinidadian bats. That is really different from where we normally see these viruses. Normally, we see filoviruses in sub-Saharan Africa. We see Henipa viruses in Oceania and Southeast Asia, as well as filovirus reactive antibodies in humans and bats in Northeast India imply zoonotic spillover. And there are many disease screening studies that show that bats are making these immunoglobulins, but no one's really delved into what those look like and what's going on with that immune system. Okay, so a little refresher on immunoglobulins and looking a little bit at the participant list. This may be review for some people, but it's always good to get people on the same page. So immunoglobulins look like a Y. This always cracks people up when I do this, but my hands are the Y. The variable region is the tip of that Y, and that is what determines a specific pathogen recognition. And if you think, so I'm going to do what I call B-cell immunology for evolutionary ecologists. So that end of the Y is a variable region. It's made up of 3 genes. Where those 3 genes, the V, D, and J, come together is the CDR3, and it's that's mainly what determines antigen binding, is this VDJ region, which is called the CDR3, or the complementarity determining region 3. The other thing you need to know is that immunoglobulins can switch constant regions, but they do so in one direction. They actually excise out the constant regions in between. So if an antibody will start out expressing IgM, but then if it needs to switch to IgA, it's going to cut out all of the intervening isotypes actually physically in that germline. So if it switches from M to A1, it can go to G4. These are the human isotypes, but it can never go back to G3. Okay, so bringing this all together, diversity in immunoglobulins comes from a series of processes, which is really useful for us because if you think about the size of a genome, a human genome is about 3 gigabases. Bats are smaller, they're closer to 2. The variable region of an antibody is coded for by about like 500, 600 base pairs. There's no way we can hard code that much diversity into our genome. Our genome would be all immunoglobulins. We wouldn't have room for actin. I'm not a cell biologist, but I've heard actin is important. So the way that humans and many other species have gotten around this is by having these suites of V's, D's, and J genes, which join together in a given BCR and create that diversity through just the combinatorial diversity, as well as insertion, deletion, mutations at those junctions. That happens for humans in the bone marrow, for birds and other species in the bursa. I think I saw Jim Kaufman on there, and I'm trying not to embarrass myself, which creates a naive B cell. And then once these cells have encountered an antigen, they can undergo further mutation, somatic hypermutation, as well as clonal expansion and selection to find better binding antibodies, and also undergo this class switch recombination. So the question is, it seems like bat innate immunity might be special, but is bat adaptive immunity special? So the 2 main questions I'm going to focus on here are, is bat B-cell immunity like other mammals? And also, is B-cell immunity similar between bat species? Because I mentioned there are over 1,400 species of bats. There are actually almost 1,500 species of bats. I think last count was 1,491. So that's a lot of diversity, and we can't really treat them just as this one monolithic thing. And here are some gratuitous bat photos. This one is really fun. This is Thyroptera tricolor, or a spix disc wingbat. They have these little discs on their wrists and their ankles that allow them to, they're little suction cups that allow them to stick in the leaves, because they roost in these rolled up leaves. And then this one is Vampirum spectrum, which is the largest carnivorous bat in the world. It's found in Central and South America. And this one even eats other bats. And I actually caught that one. I took this photo. Okay. All right. So in terms of is bat B cell immunity like other mammals, I'm going to break it into these 4 sub-questions. So first, is germline diversity similar? Are the expressed repertoire similar? Do bat antibodies undergo somatic hypermutation? Do they have these processes that we recognize from other animals? And is there evidence of antibody selection? Or do bats really not need their adaptive immune systems because they've got this fantastic innate immune system? So this is changing now, but for a long time, little was known about the germline IG locus in bats. And honestly, there's like me and a couple of other people who are really interested in this. But we did have some data. And what these 2 studies, for example, showed was that there really wasn't a consensus. So the little brown bat, which is Myotis lucificus, this is a very common bat found across North America. If you've heard about declines from white nose syndrome, you're thinking mainly about the little brown bat. And this study went and looked at did some PCRing of the genomic DNA, as well as looked a little bit at expressed repertoires, and estimated 236 germline genes in the heavy chain V3 family alone. They looked at 67 rearranged sequences, and they determined that they didn't find that much. They thought that there was not much somatic hypermutation. A study in a very different and distantly related bats. So bats, those 1,500 species, are split into 2 large suborders. One is the Enterochiroptera, which used to be called the megabats, and there's also a few other species. And then there's the Yangochiroptera, which form most of what used to be known as microbats. So Myotis lucificus is a microbat. Pteropus electo, pardon me, is a megabat. And so from this one, these people estimated that they had 51 possibly functional germline genes based on the genome. And they looked at some rearranged sequences, but they didn't quantify somatic hypermutation. So not a lot known, a lot of questions, not a lot of answers. So let's first start by looking at the germline. So to look at this question, in my postdoc, I analyzed the expressed B cell repertoire of 8 species of bats. So here are some lovely headshots. The majority of them come from this family called the Philostomidae, the American leaf-nosed bats. Desmodus rotundus here is our common vampire bat, a little cutie there. This Artivius jamaicensis will return to as Jamaican fruit bat. Most of these guys eat fruit, although these 2 eat nectar. And then these 2 are vesper bats. So Aptescus fuscus is the big brown bat. It's found all over North America, and Myotis californicus is a little small insectivore that lives in California. Okay, so do their germlines look like other animals that we recognize? They do. So if we look at the germline V genes, bats are expressing a lot of basically all the canonical IGHV families that we recognize from humans and other species. So humans are in black here. This is just a cladogram showing where things that are closer together, have fewer nodes in between them, are more closely related. And you can see that bats have retained all of the major human V families. They do have V5 and V6, they're just not shown, as well as some V gene families that are closer to stuff found in sheep. So yeah, and this is not totally surprising. These V families split before bats split from other mammals, but it does show that bats have retained them. In terms of what their CDRH3 lengths look like, they kind of fall between mice and humans, where we have the most data. So in general, humans have a little bit longer CDR3s than mice. Bats are kind of falling in the middle. You can see there's a little bit of difference in what is the most common CDR3 length between the species, but they're all kind of falling in the middle there. But it's not just retention of these ancestral genes and very similar CDR3 lengths. We do see some bat-specific species-specific V gene diversification. So here I'm showing you from this repertoire-seq data, this is a subset of the cladogram from the V3 family genes. And you can see clades where, it's all red. So, Desmodus rotundus had a species-specific diversification of this particular gene, or, blue's grouping with blue, purple's grouping with purple. But you can see so there is some species-specific diversification, and that allowed me, using this repertoire sequencing data, to estimate this number of germline genes. So this is sort of on the order of what we see in humans. It's, high 30s for most of them. I was being quite conservative with this, so we didn't have germline references for most of these, and we still don't for most of them. So in order to infer what I thought was a germline gene, I was making sure that I was finding identical V gene amino acid sequences and at least 2 different CDR3s in IgMs, which I would expect to be the least mutated. But all this seems sort of similar, at least roughly similar to what we see in humans. So is germline diversity similar? Yeah, I mean, they've got the same sorts of V genes. They've got similar CDR3 lengths, probably the same number of roughly the same number of germline genes. So yeah, it's seeming like it's pretty similar. What about, do they undergo somatic hypermutation? Do we see any evidence of selection? Because that previous paper had said, well, it seems like they have these really, really huge germline genes and no somatic hypermutation. But we actually do find a lot of evidence for somatic hypermutation. So if I look for clones that are only found in IgM, clones that are only found in IgG or IgA, one of the switched isotypes, or things that are found in both, I find actually the majority of the sequences that we were looking at were found only in IgA or IgG, suggesting that there is some mutation. Because if there wasn't mutation, we would expect that we would see these same sequences represented in the IgM. Okay, so yeah, they do undergo somatic hypermutation. In terms of, is there any evidence of antibody selection? We can look at V family usage and say that yes, in different isotypes, so everything starts out in IgM, switches to IgG or IgA or IgE. Most of these bats do not have IgD. That's not super uncommon. Some of them do have IgD, but we didn't quantify it, and IgE is so rare that at least in this analysis, we didn't include it. But you can see that, yeah, these V families are not used in the same proportion in all of these populations of isotypes. In particular, just pulling out one thing, you can see that a number of these species are using V7 a lot more in their IgA repertoire than in their IgM. So this suggests that there is some selection going on, that, something is happening that is causing certain V genes to be more prevalent in be used more with certain isotypes than others. Okay, so they undergo somatic hypermutation. There is evidence of antibody selection. So looking back at this, maybe this Taropis study was right, 51 possible functional germline genes, but it's seeming less likely that we have this situation where bats have this really, really huge germline and very little somatic hypermutation. So in answer to the question, is bat adaptive immunity special? I mean, bats generate highly diverse repertoires through similar mechanisms to other mammals. There's some bat-specific evolution with their own sort of diversification of genes, but that's kind of what we would expect with these long evolutionary timescales. Nothing super crazy. So I was ready to say, not that special. Like, adaptive immunity itself is special. B-cell receptor production is like B-cell receptor generation, and the diversity is special, but bats don't seem to be doing anything crazy unique. Okay, so that was great, but then a pandemic happened as I was going to sort of try and write this up. In the meantime, I started my lab. I had 2 children. I'm showing this picture because it cracks up my friend. She says that I gave birth to the same baby twice, which maybe I did. I'm a California native, so encountering my first hurricane was fun. This cracked me up. In Hurricane Ida, the tower that brought all of the power into New Orleans fell into the Mississippi River. But before it did, this is what the outage map was showing because the French Quarter has the unique attribute that all of the power lines are underground there versus being above ground in the rest of the city. So for the rest of the hurricane, it looked like Bourbon Street was still partying, even though the rest of the city had lost power, as well as lab delays. And in the meantime, PacBio got a lot cheaper, and these long read sequencing technologies got a lot better. So I thought, why not go back and see if this actually holds? So we're going to go back to these same questions now with better germlines, as if we were breakfast cereal, now with more raisins. So I'm not going to look so much at the express repertoire, but we're going to go back to, do bat antibodies undergo somatic hypermutation? Now that we can get a good germline reference, we can actually estimate that better. Do we see evidence of antibody selection? And then is the germline diversity similar? Okay, so this is a study that was led by Ashley Rears, who's a postdoc in my lab. It's up on bioRxiv if you're interested. And this was a study in mostly captive Archibius jamaicensis, these Jamaican fruit bats. And yeah, bats do undergo somatic hypermutation. Not as much as with people, but they do. So in the blue and the pink, you have 2 uninfected bats in the dark blue, 2 rabies-infected bats in the light blue, and a wild-caught bat that I caught in Costa Rica. And you can see in both their IgM and their IgG, they definitely do have somatic hypermutation, these replacement mutations, but they don't seem to do it quite at the level of humans. So all of these gray things are 3 different human studies. And you can see by and large, there are lower levels of somatic hypermutation, but they are there. Incidentally, a technical note, your reference really matters for doing the estimates. So we looked at estimating somatic hypermutation in these repertoires using a human reference, Aptescus fuscus, which is the big brown bat over here, separated by 51 million years from the Archibius jamaicensis, and then a genome-specific reference. And also just to give a sense for what this means, because I don't know about you, I don't have an intuitive sense of how different things are that had a most recent common ancestor 51 million years ago. The difference between humans, Ashley in this case, and a squirrel monkey or any South American monkey is about 43 million years ago. So it's pretty diverged. And what you can see here is that not only does using a species-specific reference give you much lower, or rather using a non-species specific reference gives you artificially high mutation estimates. You can also miss out on the increase in somatic hypermutation with rabies infection, especially in humans. We see that it actually estimates a decrease in somatic hypermutation. So unfortunately for me, who's interested in all of these fun species that don't necessarily have good references, it turns out that the reference actually is really important. And we do see evidence of antibody selection in response to infection. Not only did we see increases in somatic hypermutation when these bats got rabies, but we also see a decrease in their Shannon diversity, suggesting probably that there's a dominant clone or dominant clones that are really taking over after that infection. Okay, so going back to this, do they undergo somatic hypermutation now that we have better germlines? Yeah, they do. I'm gonna lessen my critique of the study that was like, oh, they undergo really low levels of somatic hypermutation, because it does seem like they undergo lower levels of or I say somatic hypermutation, I mean just mutation in general, I apologize. But they do, and we do see increased levels of mutation after rabies infection. So they definitely are undergoing this process. Is there evidence of antibody selection? Yeah, seems like there is. So again, this all seems kind of similar. And now we look with better germline references at whether the germline diversity looks similar. Yeah, it kind of does. Or it might even be simpler than humans. So here's the Jamaican fruit bat. You can see a number of V genes, Ds, Js, and Cs, but this all looks kind of similar to what we expect in humans, or even simpler, because humans have had a lot of duplications within their constant regions that have led to 4 IgG subtypes, 2 IgA subtypes. IgE had 1 pseudogene. So, and in bats, we're just seeing at least in the Jamaican fruit bat, we're seeing a single IgM, a single IgG, a single IgE, a single IgA. As I told you, some of these bats don't have IgD. This is one of the ones that doesn't. And similar levels of V genes to what we see in people. So Artibis jamaicensis weirdly has a lot of pseudogenes, but they have 57 functional genes. This is roughly similar to humans, 40-ish. And if we look at Aptescus fuscus, that big brown bat that I was showing you that we used the reference for a few slides ago, similar. There's a bunch of V genes, we see a bunch of D genes, J genes, and then again, a single IgM. These ones do have IgD, an IgG, E, and A. Okay. And so going back with the germline references and actually counting the number of functional V genes in those genome references, as I told you before with the RepSeq, I was being kind of conservative. So I actually estimated about half of these genes. So in reality, there are about 62 if you include open reading frames for Artibis jamaicensis. There are about 75 for Aptescus fuscus. So, okay, definitely underestimated it, but we're still not seeing that, 230 V3 genes alone. So is the germline diversity similar? At least in these two species, seems like, yeah. I mean, we underestimated it, and it's good information, but it's not something strikingly crazy. One thing that is a little bit weird is that it seems like an entire suborder has just ditched the kappa locus. So in RepSeq studies as well as germline references, it seems like the Enterochiroptera kept their IG kappa locus. Yangochiroptera, we just don't see any evidence of it. In fact, when we try and find it in Aptescus fuscus, this is a study led by Taylor Purcell, we find like 2 J genes, one of which has an RSS sequence, 3 J genes rather, one of which has an RSS sequence, but it looks like it's gone, which is very interesting, and we're very excited to follow up on it. But this is not unheard of in terrestrial vertebrates. Chickens have also lost their kappa. Got a really fancy looking chicken. Okay, so they're kind of special, not the most special. And I was ready to sort of say, like, yeah, bats, they do things that we are really familiar with. Everything, like it's bat specific, but it's not crazy. And then Aptescus fuscus came and Kool-Aid manned his way into our study. And so it turns out I am wrong. Some bats are actually very special. So this is work that was led primarily by Taylor, assisted a bit by Ashley, and then the experimental stuff was done by James Ellison. And we also, and as I said, Taylor is a student in Scott's lab, and this was stuff I started when I was a postdoc with him. So I said the bats look similar, and they do. We've got this very recognizable V's, D's, J's, and then the constant regions, except also not, because it turns out that Aptescus has an entire second functional heavy chain locus. That's actually even bigger than the one I showed you before. And this is kind of weird because generally vertebrates don't have 2 heavy chains. The only other time we've seen this in vertebrates is in teleost fish, so in things like salmon and trout, where it has happened in a completely different way. So that is actually kind of weird. Here we have a mammal with 2 separate heavy chains, and it looks like from doing single-cell RNA sequencing of this that these different loci might actually have different functions. So we call them locus A and B, A is smaller, it has a much higher level of mutation, and it's much more used, especially in the naive repertoire. Whereas IGHB is a lot bigger. It's the only one that has, for example, V7 and V6 genes. It doesn't seem to mutate that much. It's not used as much, but it's where it is used is mostly in antigen experience differentiated B cells. So you can see most cells are using the A locus, except for these non-class switched antibody secreting cells. And this is showing you just the percentage of somatic hypermutation. And you can see that especially in class switched B cells and class switched antibody secreting cells, you see a much greater level of somatic hypermutation in locus A, which suggests that in addition, that bats have not only that these bats not only have sort of their innate immunity and their adaptive immunity, but their adaptive immunity is sort of functionally differentiated so that they have one locus that's kind of ready to go, that B that's less used, but is more likely to just class switch and start secreting antibodies. And then one that is sort of more trainable and even more adaptive with the IGHA locus. So in my kind of silly castle analogy, it's almost as if instead of just having, these normal B cells back here, there's actually a functional separation between these two loci where IGHB is serving as like a diverse standing army of antibody producing cells ready to go. And then IGHA are these like wealth of highly trained assassins, things that can be even more specialized. And thank you, Internet. If you Google assassins, you can get this lovely set of pop culture assassins. And I was particularly pleased about the lady representation. Okay, so is that B cell immunity like other mammals? At least for some, it's actually not special, it's extra special. This is super weird and cool. So then the question is, did we get lucky? Because actually we only looked at this one Vesper species, Aptescus fuscus. It happened that our collaborators at the CDC were running a vaccination study on them because they're really common in North America. There are almost 1,500 species of bats. Did we just stumble on the one that has this? It turns out we did not, sorry, got confused with my slides a little bit. It turns out we were not just lucky. So then there's this question of, is B cell immunity similar between bat species? And it turns out that actually we were sort of lucky, but we weren't that lucky because this duplication of the locus from the V genes through at least IgM seemed to be duplicated at the base of Vespertilionidae. So here is a phylogeny of the species that we looked at their genomes for, and then this is the IGHM constant regions, mostly the CH1 for these species. And you can see this kind of perfect bifurcation where it looks like at the base of Vespertilionidae or shortly before it split from other bats, there was this duplication of the locus. I say through IGHM just because if you look into the end, certainly this entire locus got duplicated, but there's a lot of gene conversion that makes the relationships a little bit hairy. So it turns out it looks like it's in this whole family, with the exception of there's one species where we don't seem to find it. And that's really interesting to me as an evolutionary biologist and ecologist because Vespertilionidae is actually is the largest family of bats. There are 531 species. They are basically a third of all bat diversity. They're also half of the diversity of Yangochiroptera, that larger of the two suborders. And so to me, it makes me wonder, and actually when I gave you this diversity slide before, all of these are actually Vespertilionid bats. And it makes me wonder if this duplication of the heavy chain locus might be instrumental in their evolutionary success and their ability to be on every continent except for Antarctica. Yes, every continent for Antarctica. They don't get to New Zealand. That's its own special family. And we also see adaptive duplications in Vespers in protein kinase R, which seems to me suggest to me that there's something interesting going on with these guys. So to further look at this, this is stuff that is very much still in progress, but an effort that's being spearheaded by Ashley to look at, is this B cell immunity similar between bat species? So with the humans and the we've looked at humans, Artivius and Aptescus in great detail in this talk. And what we see is that there's actually kind of they're similar in terms of the number of V genes, not the same. Artivius had 57 functional V genes, Aptescus had 75. Humans have about 50. But we're not really seeing this 236 V3 genes that Sarah Bratch and her colleagues were finding in their Myotis lucificus. So we thought, is this even possible? Maybe there's diversity that we're missing just by looking at these, couple of species. And it turns out that it is completely possible because as we've been looking at more and more species and quantifying the number of V genes, we're finding some that have huge numbers of genes. So actually, looking at Myotis humanensis, which is not the same species of Myotis, but a congeneric, we find 231 functional genes. So that 236 is actually not looking crazy at all. And we find some truly spectacular diversity in the number of V genes within bats, where molossus has a total of 10 IGHV genes, whereas Placotis auritus has 708. And even if we filter out the functional genes, it's filter 2 functional genes, it's still 7 versus 588, which to me is crazy. It seems like there's probably also a lot of interesting diversity structurally in terms of like the size of the locus. So Artivius jamaicensis is insanely large. It's like 7 megabases, whereas other ones have, on the order of kilobase sizes. When we look at the dual locus ones, we have bats with different orientations on the chromosomes and different sizes, as well as sort of different levels of, some have V genes that are going in the opposite direction. Some have some V genes in the forward and some in the reverse, like we see in the human kappa locus. So we're excited to dive into this, but it seems like B cell immunity is really not similar between bat species. So that'll be an exciting thing to see what are some of the correlates or possible drivers of this diversity. So returning to our question, is bat adaptive immunity special? I would say bats are all unique and have their own special place in the puzzle of the universe. Definitely can't treat these guys as a monolith, and definitely, to me at least, are special. So Ashley and I will continue our quest for world domination by looking at more and more bat immunoglobulin repertoires. I thank you for your attention. I really, again, I want to thank especially Taylor and Ashley, but Tony Shounds, who did all of the Artivius jamaicensis lab work, James Ellison, who did all of the infections with Aptescus fuscus, Scott Boyd, who's been an incredibly supportive mentor and collaborator, a number of funding sources, and I am happy to take questions. So weird not being able to see anyone. Yeah. I don't know how to call on Jim, but
We have a question in the Q&A from Raj. I don't know if you the Q&A. Raj says, IG neutralizes viruses, opsonizing pathogens for phagocytes, and activates complement to destroy pathogens. How might these antiviral defenses contribute to preventing viruses from being cleared in bats, allowing bats to serve as natural reservoirs?
yeah, it's a great question. I think if you talk to Taylor, she's found some really interesting evidence. It seems it, and she has an interesting hypothesis that IgM actually might be more important and the sort of main functional isotype for bats rather than IgG. And so in that way, there may be sort of, if there's less reliance on IgG, maybe there's a little bit less clearance. But the idea of, exactly what tolerance looks like in a bat and how they are able, there are a lot, I feel like every time we find something cool, we just end up with more questions because it's like, okay, cool. They're not having a crazy inflammatory response. But then how are they controlling virus in the absence of this response? And so yeah. Do bat species with more V genes also have other specialized immune-related genes, such as those in MHC? Um, we don't know yet. I'm one of the things I'm excited to look at is, whether there are correlates between MHC and V gene stuff. There was a great paper out by Yana Safanova and Corey Watson was on it. And they were showing that there is, and Matt Pinnell, they were showing some evidence of a trade-off between like things with larger heavy chain repertoires seem to have smaller light chain repertoires. So there might be a little bit of a trade-off there. Um, do bad NK cells have FC receptors for IgG so they can mediate ADCC? Uh, Great question. I don't know off the top of my head. I have a grad student who's actually looking at FC receptors. Um, there's not amazing data. Like it's hard because we don't have antibodies. So a lot of the traditional cell biology ways of finding these things out are a little bit tough. I could ask Taylor. Taylor has been looking more at our like single cell RNA-seq stuff, and she could see if there are the FC receptors expressed on them. But a number of the FC receptors that we recognize and FC receptor-like proteins that we recognize from other species have been lost in certain species of bats. And that's something that one of my grad students is jumping into. Um, okay. Oh, thank you. Jim.
And you can talk.
Hi, can you hear me?
I can, yes.
Oh, great. Thanks. What a fabulous talk. Thank you so much for all the really new information. I knew none of this. I have two questions, actually.
Yes.
First is you mentioned that the only other group of animals that has duplicated IG loci are the teleos fish. And I guess you would know as well as anyone that
IG heavy locus, I think.
yeah, so sorry. But they've undergone a genome-wide duplication compared to most other vertebrates. And actually, there's another system where this has been seen. Louis Dupasquier has done a lot of work with polyploid frogs. And there's a whole series of frogs which go from essentially being diploids to dodecaploids. And in general, they keep all of their IG loci and diplodize their MHC. So this is a really old story from him. And so the question is there any evidence for this kind of genome-wide duplication in the particular bat species? This, I forgot what you called it.
Vespertilionidae.
Yeah, it goes by too fast. But whatever that is, that begins with a V. Has anybody looked at the whole genome? Because you also mentioned adaptive duplications of protein kinase R.
Yeah, it's a great question. So I can tell you that their genomes are not like they're so I don't know if the degree to which people have found whole genome duplications. I know that their genomes are roughly the same size as other bats. So certainly they haven't retained both copies. But whether this has come from an ancient genome duplication that like then they've lost most of what was duplicated, I don't know, but it's a really great question, and now I want to look.
Yeah, I think it's a fascinating possibility. The other question I had is I got a bit confused between the two loci that you have, the small one that undergoes somatic hypermutation and the large locus which does not. But then I thought I understood that the small one that undergoes somatic hypermutation is found mostly in naive B cells, which shouldn't be doing somatic hypermutation, and that the one that doesn't undergo somatic hypermutation so much, the large locus, is primarily in those B cells which are differentiated. So I just couldn't quite fit that together in my mind.
Yeah. Yeah, it's it also took me a little bit to wrap my head around. So it seems like most cells seem to use the small one that can undergo somatic hypermutation. Where we seem to see the most B cells with the most use of B is in the non-class switched antibody secreting cells. So things that went like, IgM secreting plasma cells.
Got you. So that's what CS means. I'm sorry, I didn't know what that meant.
Sorry, yes, class switched.
Got
So
you.
it seems like you have some, you know, the ones that express the B locus seem to quickly switch to an antibody secreting thing without undergoing much mutation or even class switching.
So these couldn't be like B1 B cells or something like that.
Um, I don't know. So that is one thing I've been meaning to uh, look up. I picked up immunology as a postdoc, and I am still learning.
Well, that goes for all of us, no matter how long we've been working on it. But if you've been doing single cell RNA-seq, then you will know about IgD expression and things like that. And I
Yeah.
think you mentioned that IgD has been dumped in some bats. Does IgD presence or absence map onto these duplicated versus non-duplicated bats? I didn't follow fast enough.
No, totally. I I would need to check back. Especially Taylor's been the one who's been spearheading looking at most of the single cell RNA-seq. So the short answer is I don't know. I think we mainly looked at IgM. I can't remember how much IgD is expressed, but
Yeah, but the data may be there, which you
yeah,
can look at. It was the
yeah.
Herzenbergs in Stanford, where you were, who first came up with this B1, B2, and it was based on IgD, and I've forgotten the details. But perhaps this is one way to approach this.
Yeah. No, that's a fantastic suggestion. Thank you.
Anyway, fabulous talk. Thank you so much.
I appreciate it. Okay, was there another? Oh, can you elaborate more about the PacBio protocol used to do bat germline sequencing? Yes. So some of these things have come from, there's a large genomic consortium called BAT1K, which has been doing a lot of these genomes, and they've done, so that's where our Aptascus fuscus reference came from, and that is a combination of PacBio hi-fi reads, Hi-C, and I think that's mainly what they're doing now. So just like enough coverage to get really nice big contigs and then the Hi-C to help figure out like the orientation of everything. We, for our Archimedes Chimay census, we just did PacBio Hi-Fi like 30x. So long read sequencing. We used fifth passage fibroblast cell culture just to get a lot of it. And that was done at the University of Louisville, someone that Corey Watson connected us to. So, and that worked, like it worked well enough that basically just getting like long enough contigs, our heavy chain locus was on a single config and our lambda was over 2, but worked pretty well for us to get it.
All right, we're at the top of the hour. If there are no other questions, I would like to thank Hannah for this fantastic presentation. Thank all of you for attending the webinar, and I hope you all have increased respect and admiration for our bat cousins. If you want to review this webinar, it'll be made available on the Society for Immune Polymorphism's YouTube page. Our next webinar will be on September 30th. Doctors Melissa Smith and Corey Watson from the University of Louisville, which have come up several times in Hannah's talk, will be presenting on the genes that encode the T cell receptors and the B cell receptors. And if you joined late, I want to let you know that the Society's second annual symposium in Sassenheim, the Netherlands, will be held from November 3rd to 5th in 2025. focusing on polymorphic immune molecules and their functional diversity across species. Early bird registration ends on August 15th. You can register and find more information by visiting sip-sassenheim.org and immunepolymorphismsociety.org. We're looking forward to seeing you in September.
And you can feel free to email me. If you just Google Hannah Frank Tulane, you'll find me.
All right, thank you, everyone.
Thank you all.